Hello, everyone, and welcome to today's live broadcast. Tough targets, simple genotyping, fast and easy protocols to determine SMN1 and SMN2 copy number and APOE allele identification. Presented by Ryan Donnelly, Product Manager, Canon BioMedical. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Canon BioMedical. For more information about our sponsor, visit www.canon-biomedical.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during this presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask Question box and let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Ryan Donnelly. I will now turn the presentation over to him.
Thanks for that introduction, Kaylee. We're excited to bring to you today some really quick and simple protocols for some traditionally difficult to analyze targets, specifically copy number changes related to the SMN1 and SMN2 genes, as well as how you can go about investigating common SNPs in the APOE gene. Some quick disclaimers before we get into the meat of the presentation. The products that you're going to see here related to the Novallele line as products of Canon BioMedical are all for research use only, not for use in diagnostic procedures. If you need to find out any licensing information or product-specific information, you can find those in our user guides for the specific products that we're going to list here online at Canon BioMedical, www.canon/biomedical.com, or from a Canon representative. We, as Canon BioMedical, support the products directly in the U.S. If you're in the UK or Ireland, we have a distributor through LabTech. Canada is through Cedar Lane. The Benelux region is supported through SanBio, and the DACH region, which includes Switzerland, Austria, and Germany, is supported through Biozol. And the last thing to touch on here is Novallele is a trademark of Canon BioMedical. So now that we got that out of the way, we can start the actual presentation. From an agenda perspective, we wanted to outline Canon and genetics, a little bit of background on Canon BioMedical, then get into selecting genotyping technologies for difficult to analyze targets. So what type of considerations go into choosing a method for these investigations? And then the next two sections will be related to applications that we support through the Novallele product line. So the first one is we will look through our copy number solution for SMN1 and 2 copy number determination. We'll look at the method and some data analysis for those. And then similarly, we will run through the method and data analysis for SNP genotyping example. In this presentation, we're going to use the APOE gene to show that example. Rounding out the end of the presentation, we have an analysis software that we'll walk through. That's the Novallele HRM Analyzer software. We can show you how to use that. And lastly, kind of how would you go about finding more information about these products if you so desire. So these seem like a solution that you want to try out in your lab. How do you go about finding these products on the Canon site? So first things first, Canon and genetics. You may be wondering why we're here. You may be wondering if we have cameras or microscopes for your life science research. We get that question a lot, and the answer is actually no. So we're taking a step adjacent to some of the more imaging-focused technologies that Canon is well known for worldwide. Canon BioMedical focuses on genotyping chemistries, so chemistries for SNP insertion deletion testing, as well as copy number identification. And that's where Canon BioMedical is focused. Canon BioMedical was launched in 2015. It's a subsidiary of Canon USA. The desire here was really to go beyond the adjacent companies, subsidiaries that Canon has that do some work in digital imaging and digital X-ray and push further into the biomedical field. So that's where Canon BioMedical was launched out of. So as I mentioned, we launched in 2015. Toward the end of 2015, we launched the first products for Canon BioMedical. These were genotyping assays and reagents specifically for SNP mutations, insertions, deletions. We have some mitochondrial DNA targets as well. The primary focus area for these is in neuroscience research and pharmacogenetics. This product line will be the example that we touch on when we look into the APOE mutations from the protocol example. We expanded beyond that in November 2016. Listening to customers, one of their desires was to have characterized controls for the assays. So we ended up launching control sets for each of the genotyping assays in November of 2016. So for all but a few of the genotyping targets that we have a matched control set for those, which is really important when you get into more rare mutations. And lastly, late last year, October in 2017, we rolled out copy number assays. So we had heard from the market, looking for a simple and quick method to investigate copy number changes in SMN1 and 2. The technique that we use, we'll get into a little bit later, high resolution melting, allows us to kind of bring in this application. It goes well beyond the simple SNP and indel detection that we had initially launched with, and it allowed us to open up this new application. So the next thing we wanted to touch on, just kind of go over kind of background on the methods available if you're not doing investigations in your lab. So what type of genotyping technologies are out there. And we can give a little bit of an overview around the Navallele method. So there's many different types of research. So we've broken it out into a few different areas. So from a pre-screening perspective, what we mean here is kind of inclusion or exclusion criteria for sequencing studies. So the thought here is, instead of sending all samples for sequencing, if you can pick up common mutations that exist within them, you would likely have to sequence fewer of your samples. The next, into a true screening perspective, when we say screening, our thought here is really, we see a lot of interest around CRISPR clone generation. So using genotyping in lieu of Sanger sequencing to pick out the clones that have picked up either the knockout or the insert from your CRISPR studies. And lastly, verification. So we see a lot of discovery work, kind of basic research around SNP arrays. So the thought there is, if you pick up a handful of SNPs on the SNP array, you can use a targeted genotyping approach as a parallel method to make sure that that SNP actually exists within the sample. And across all these different types of research, there are different mutations that can exist. So we've talked a little bit about SNPs, insertions and deletions. Copy number variations can exist. There are arrays that pick up kind of global copy number changes. Methylation signatures can change. And also, somatic mutations, which are heterogeneous through samples, are also things that can pop up. So when you're looking at options, and you lock down the application that you're going after, then the technology options start bubbling up to the surface, and we can try to pick which one makes the most sense. So there's many different methods out there, from next generation sequencing, which is really kind of one of the ultimate discovery tools, to Sanger sequencing, which is a gold standard for mutation detection, hydrolysis probe PCR. There are multiple millions of SNP assays around these hydrolysis probe-based PCR, great for targeted approach for single-plex assays. And then high-resolution melting analysis, which the Novallele line is really based around. And while it's a lesser-known technology, there are many, many applications that are supported by this technique. So in essence, it is an endpoint PCR that is followed by melting the product. So it's a very simple and quick protocol, but it has many applications that go beyond what some of the more targeted approach like hydrolysis probe PCR can pick up. So once you have the application and the mutation type, it really can drive the method selection for your genotyping studies. To touch a little bit on the basics of high-resolution melting, as I had mentioned, it is a standard endpoint PCR followed by a melt at the end, so to melt the product. What we use is a intercalating dye. So the dye is not attached to a probe, unlike some of the hydrolysis probe methods that we had touched on. So it's non-target specific. It's an intercalating dye. It sits in between double-stranded DNA. So at the end of the PCR cycling, you have the maximum fluorescence because you have the most double-stranded DNA available. A melt occurs at a fixed rate, and throughout the entirety of the melt, fluorescence is monitored. So you're watching the decay of fluorescence. And it is this decay in fluorescence which gives you a signature in which you're able to pull out if mutations are present, if copy number changes are present within the sample. There's many features and benefits of high-resolution melting. The first thing I'll touch on is really just kind of what it looks like. So in the top left graph here, you see 3 different species of samples. So the black curve is the wild type, so non-mutated. The red curve is a homozygous mutant sample, so both alleles are mutated. And what you see here is just they both have a similar shape. It's one melt domain, and you just see a shift in the temperature in which it melts. And that makes sense. Well, the wild type has 2 alleles that are non-mutated. The homozygous mutant has 2 alleles that are mutated. So it's within that sample, it is one species exists. So that's why you just see a change in the temperature in which they melt. And that's different from the heterozygous mutation, where you have one allele that is non-mutated and one allele that is mutated. So in that one, you have 2 species that exist. So not only will you see a temperature shift, but you also see 2 different melt domains. And that's why you'll, in this example, you see kind of 2 different melt transitions there in the blue curve. But from a benefits perspective, the method is quite accurate. All of the Novallele genotyping and copy number assays are thoroughly bench tested for consistent results. It's quite cost effective, as I mentioned. It is it's a fluorescent dye. It's not a fluorescent probe, so it's from a chemistry perspective, it's quite cost effective. It's very fast. It's a standard PCR followed by a melt. Results in a few minutes after your PCR is completed. It's very simple. So it really only requires a high-resolution melting compatible and enabled thermocycler. And as I alluded to in the previous slide about applications, it's very versatile. So we can pick up SNPs, insertions, deletions, very large deletions, which are interesting, and copy number changes. And those are specific to what Novallele covers. But going beyond that, high-resolution melting can be used to investigate methylation signatures, mutation scanning, and even triplet repeats. So I kind of touched on this already, kind of the target types that Novallele supports, SNPs that we'll touch on for the APOE, mutations, copy number variations. This will be the SMN1 and 2 example. Mitochondrial DNA, we have targets available for that. Small and moderate insertions and deletions, which are pretty common for genotyping investigations. So these there are other parallel methods like hydrolysis probe that can pick these up. But for large insertions and deletions, those those don't show up in the menus for those hydrolysis probe PCRs. So with Novallele, we actually have assays that pick up to a 7.6 kb deletion in the hexagene. So this allows us to really cast the net wide when we're talking about insertions and deletions and what size of those we can pick up with the technology. Specifically for Novallele, it's a very fast and simple protocol. It requires extracted DNA to be mixed with the specific assay of interest along with the Novallele master mix. So upstream of plating, the DNA is extracted with any really commercial available DNA extraction kit. The reactions are mixed up with the template DNA, the assay of interest, and then the master mix. Those are plated and then dropped onto a HRM compatible thermocycler where the PCR and melt take place, and then the results can be either analyzed on the native software available to the thermocycler, or they can be analyzed using the Novallele HRM analyzer software, which we will go into in some more detail towards the end of the presentation. So when I mentioned that you'll need an HRM-compatible thermocycler to do the genotyping or copy number investigations with the Novallele line, you might be asking, well, what are those? What companies offer those? There are many that are available. A lot of the real-time PCR instrumentation have this capability. So we do a bulk of our development work on the Bio-Rad CFX systems in-house. We've done cross-platform testing on Roche systems, so the various light cyclers, the LightCycler 2 or the LC480. QIAGEN has a system that we have that we do testing on the Rotogen Q. Thermo Fisher with the Quant Studio has a melting module that you can add. BioFire has an older system, the LS32, and the Cole Parmer Eco48. So we've seen similar data across all these systems. We have all these ones listed in-house. We've done a lot of testing on them. So yeah, similar data across them. And having the ability that many instruments have this high-resolution melting application kind of built in, you might actually have one of these in your lab or down the hall at one of your colleagues' labs. So that'll be it from a general overview of the Novallele line. What we wanted to really spend a bulk of the time on for the webinar was around the two applications, the first being copy number variation analysis of the SMN1 and 2 genes. So that's where we're going to start. And when we talk about copy number variation, I run into this a lot at trade shows, talking to individuals or just talking to potential sites. Copy number variation can mean different things to different individuals. So a lot of groups think of it as kind of genome-wide copy number changes. It's very different than what we're trying to do here. So our copy number variation assays are gene-specific. We have them for SMN1 and SMN2. And so what we look at is deletions and duplications of that gene. So a normal sample would have 2 copies of SMN1. The deletions we can pick up are either heterozygous deletions, so 1 copy of SMN1 still exists, or homozygous deletions, so 0 copies of SMN1 exist in the sample. On the flip side of that, duplications, so we can pick up if there's a duplication of SMN1 in the sample, and the example here that we show is there's actually 3 copies of the SMN1 gene. So we launched assays for both SMN1 and 2, and the thought behind that is there's a lot of homology between the 2 genes, and the biology is quite interesting between the 2 of them. So from a homology perspective, there's really 1 base pair difference between the 2 genes. They both create SMN protein, but due to the 1 base pair change, in SMN2, a bulk of the protein ends up being truncated and is therefore non-functional. So a majority of the functional SMN protein comes from the SMN1 gene. So when looking to design an assay around these 2 genes, understanding the homology that exists, it really required us to go about this and make sure that we had extreme specificity between the two assays in order to not pick up non-specific amplification of the adjacent gene. There are some other kitted methods, commercial methods, available to researchers who are looking at investigating copy number changes in SMN1 and 2. Those are really digital PCR, droplet digital PCR, and MLPA. They're available from a workflow perspective. The Canon method, depending on your instrument of choice, can be done in around an hour. The Quant Studio specifically returns results in under an hour. So that would be the PCR protocol plus the MELT protocol is done in under an hour. So it's very fast. So the other methods I mentioned here, droplet digital PCR and MLPA, definitely both have advantages as well. Droplet digital PCR is great for if your research question has you trying to go about answering if there's higher copy numbers that exist and differentiating between the two. So maybe you want to understand samples have 5 versus 6 copies for SMN2. That would be a great method to go about that. And MLPA, while it can take a long time, it's highly multiplex. So you can pick up some related SNPs as well that show up in specific groups related to these genes. But if you're looking for a very simple and fast method for determining 0, 1, 2, or 3 or more copies of SMN1 and 2, Novallele is a great solution for you. That being said, I think the next logical question was, how does it work? So there's a few things to call out here. The first is we're looking at relative fluorescence. And we've set this up, we've set reactions up to be competitive. We set them up to be competitive in 2 ways. We limit the DNTPs available in the assay mix, and we also limit the cycle number. So we actually reduce the cycle number down to 25 in order to keep reactions out of the plateau phase. If we were to allow reactions to get into the plateau phase, we would lose all of the relative fluorescence differences between samples of varying copy number. Another thing to call out is the assays are set up in a duplex. So for each assay, we'll have a target amplification, so either SMN1 or SMN2, as well as a reference gene. So in this case, the housekeeping gene of known 2 copies. One of the things that we do a little bit differently, though, is you'll see the reference there in the graph on the left, the rightmost melt domain. You'll see some variability in the fluorescence of the reference, and I'll get to why that exists in a second. So first, you will see a fluorescence difference in the target amplification. So in the leftmost graph, the leftmost melt domain, you will see 4 different kind of clusters there. So a black cluster, which is pretty much flatlined, a blue cluster, a red cluster, which is above the blue, and then a green cluster, which is seen above the red cluster. So those are separated out by the more copies of the target that exists, the higher the fluorescence there. Now, what's interesting is because we've set these up to be competitive, again, from the target versus the reference, that's why you actually see the spread in the reference gene's fluorescence. So the higher number of primer binding sites in either the target or the reference allows for more amplification of that region. So the example I'll give here is since there are no copies of SMN1 in the black curve, you see kind of flatlined under the target amplification, and the reference amplification for that sample cluster, you get the highest amount of fluorescence there because there's no competition for the reference gene. Inversely, the green sample set, you see has the highest fluorescence for the target and the least fluorescence in the reference gene, because the target has more primer binding sites than what we see in the reference. So in this case, we have 3 copies of SMN1 in the green cluster, and due to the fact that the reference is a housekeeping gene of known 2 copies, it only has 2 copies available for amplification for the reference gene. So we can use this inverse relationship to our advantage in that we can really use both fluorescence signatures for both the target and the reference to separate out samples of different copy number. So in the rightmost graph, it's the same data as the left graph. It's just shown in a different way. So this is a difference plot. So you're able to select one of the clusters selected as a baseline. In this case, we use the blue curve, which is 1 copy for SMN1. We zeroed that down. And then you'll see you're actually seeing the 2 different melt domains. So the leftmost domain is the SMN1 target, the rightmost domain is the reference, and both of those pieces of data exist to separate out the samples based on copy number. And what you'll see, what you do see in the rightmost graph, the way you actually make the call is a panel of controls is run. You have the option of starting with Corel controls for 0, 1, 2, 3 copies of that specific gene. You run those on the plate with your unknown samples, and then the call is made by seeing which one of the controls each one of your samples has clustered with. So you run the panel of controls, and then based on the clustering pattern in the difference plot of the remaining samples, you can assign copy number calls to each of the samples. So the question there is, which one of these did my sample cluster with? Did it cluster with the 0 copy control? That means the sample 0 copies. Did it cluster with the 2 copy control? That means it's 2 copies. As I had mentioned when we were looking at the protocol differences, investigating higher copy number changes and picking up the differences between, say, 5 versus 6 copies of a gene is more difficult. The fluorescence change between the 2 is much smaller than, say, 2 versus 1 copies of a gene. For that, it's in reality, you should have around 50% fluorescence versus, since 1 copy versus 2 copies. When you start getting into 5 versus 6, 6 versus 7, the delta fluorescence between those samples shrinks. So we are confident for both of these assays to call out 0, 1, 2, and 3 or more copies. Above 3 copies, it gets difficult to reproducibly call samples of those higher copy number calls. So we're confident clustering everything that, 3 or more, we are very confident in making that call. You'll see an example in the SMN2 graph on the right that we actually ran a 5-copy control there. You can just see how close that starts getting. If you add a 4-copy control in there as well, which we don't have, it would be split between the 3 and 5-copy curve sets, and you can just see how quickly kind of the fluorescence differences between the samples breaks down. So we're happy with these claims, and we've done a lot of testing internally to make sure that they are very reproducible across sample types and extraction methods and instrumentation. So that brings us to the end of the copy number application, and we want to transition into a more standard genotyping application. In this example, we'll look at some SNP genotyping assays. And for this, we're going to look at the APOE gene. So APOE, long form of the name, apolipoprotein E, it's a lipoprotein, so traditionally, biology-wise, associated with many things in the cardiovascular system. So that is in existence, but also has a large, it shows up in a lot of Alzheimer's disease research as well. and that may be where it's even more well known for. The two most common SNPs in the APOE gene are shown here. So we have the C.388T2C. This relates to a cysteine to arginine change at residue 130 within the gene. You'll see another counting there where it might be the 112th amino acid residue, but that's the legacy name, so we'll focus on the ones that are bolded here. And the other SNP that we'll show is the 526C to T, and this equates to an arginine to cysteine change at residue 176. From a data perspective, it looks quite different than the copy number data that we just looked at. From a protocol perspective, though, it is very similar. So it is just a simple PCR followed by a melt. The cycle numbers are higher than what we see in copy number because we allow these reactions to plateau out to the genotyping, since we're not looking at relative fluorescence changes. But we wanted to show some sample data sets for this. So what you're seeing here, this is the C.388T to C assay. You'll see 3 different species shown here. So the wild type is the black curve set at a lower temperature melt. The blue is the homozygous mutant, shown at a higher TM. And as I had mentioned before, when you're looking at genotyping changes, the wild type and the homozygous mutant will be differentiated at the temperatures at which they melt. They show a very similar fluorescence signature, so one main melt, just separated by the temperature in which they melt at. And then you'll see a bunch of het mutants here in red. And the way those show up, you'll likely see a melt domain, one under the wild type and one under the homozygous mutant melts, and at a lower fluorescence, because you're, in reality, you have one version of each of those alleles present in the sample. One thing that is very important with high resolution melting is your wild type will not always be at a lower temperature. It can swap places with the homozygous mutant. So while it's important to differentiate the two, you can't always assume that one of those would be, you can't always assume that the wild type would be at a lower TM like it's shown here. We have assays where it's swapped, where the homozygous mutant exists at a lower temperature than the wild type. So in that case, controls are really necessary. So we had heard from our customers after we launched the assays, having characterized controls for all these assays would be a really nice thing to offer. So we went about putting a product together that answers that need. So for each of the assays, minus a couple of the assays we have available, we have around over 300 genotyping assays available. We have characterized characterized control sets. And what you get with each of these control sets is a homozygous wild type and a homozygous mutant control that you can run on your plate with your samples of interest. The nice thing is they're diluted down, ready to use at a stock concentration, at the same stock concentration that we have for the samples that get loaded on your plate, so you can use them right next to it in the same, pretty much the same type of setup. And here's what those look like. So the wild type control shown in black, you'll see clusters very closely with unknown sample 2. So the call there would be a wild type for unknown sample 2. On the other side of that, you'll see the home control is the blue curve, and the green curve, unknown sample 1, tracks very closely to that home control. To get a het call, there is the ability to mix these together. We have a blog article about how to do this coming out very soon. So check back to the Canon BioMedical blog. In lieu of that, if you have confidence in making the call just based off the dual melt peaks of a het, so long as they exist under those the melt domains that you would expect from the homozygous mutant and the wild type, you can go that route also. So as I touched on, we have over 300 different genotyping assays, around 80 of which are related to neuroscience. So we have some in the GBA gene, not the first gene that a lot of people think of related to neuroscience, but there's been some research showing up in Parkinson's disease, as well as the hexagene, which we see in relation to Tay-Sachs. So many different targets for both of those genes. For hexa specifically, we had touched on that very large deletion assay. That, as far as we know, there's no targeted method to resolve that deletion outside of the Novallele line. So we've seen a lot of data, figures between the copy number variation application and the genotyping SNP application. And the data that you've seen has been generated using the Novallele HRM analyzer software. So we want to show you a little bit about how that works. So we had put together a very intuitive, easy to use interface for individuals either currently running high-resolution melting assays or that would like to bring them into their lab using high-resolution melting. And this is kind of a screenshot of the layout. It's very powerful. There's many different ways that you can go about normalizing data and analyzing data. So we have some curve normalization that helps with normalization of fluorescence. There's a negative derivative plot. You see this in the bottom left. And that's used for genotyping, so making your genotyping calls related to SNPs and indels. And we do curve comparisons via clustering, and that's in the bottom right graph, and that is more similar to what you saw in the copy number application, so clustering the controls to the samples. So this supports both the genotyping and copy number applications with one software. To access it, the website is seen here. It's free to use. It's cloud-based. It requires a sign-in, so first name, last name, email address. Very simple to use. If you're currently running high-resolution melting assays in your lab, this is supported by a number of the more common high-resolution melting capable instruments for seamless data upload. For ones that aren't supported, we can help with kind of formatting an Excel table to get the data in here so it can be analyzed. Okay, if you already have high-resolution melting data and you're just looking for maybe a more elegant or simple to use or user-friendly analysis software, feel free to check out the Novallele HRM Analyzer software. Or if you're looking specifically for the Novallele assays, we highly recommend you go about using this. Our support team is very happy with it, and the customers that have used it have really liked it. So that really brings us to the close of our webinar here. So we have, I just wanted to relay, if there's interest in the specific applications or even the targets that we touched on today, so copy number changes in SMN1 and 2, or SNPs in APOE, I can show you how to go about finding those. Or as we spoke about, we have many, many more genotyping assays available and how to go about searching for those. So the Novallele assays in chemistry, the copy number assays are packaged for 200 reactions in each vial. These are 10 microliter reactions. Genotyping package is a little bit smaller, 166. And the controls matched to the genotyping assays, we package diluted down, ready to use 30 reactions per vial. We have different kind of focus areas here. So a bulk of the genotyping assays fall into either neuroscience or pharmacogenetics research. So we have a a very large menu of CYP-related mutations. As well, you'll see here the things that we haven't talked about, so things like inherited disease-related targets, metabolism-related targets, and cardiovascular genotyping assays are also where we have a lot of coverage across those research areas. There's a couple different ways to find an assay. So you can browse. This is what's shown on the left. So we have what's called discover zones. So things like I mentioned, cardiovascular, inherited disease, metabolism, neuroscience, pharmacogenetics. If your research takes you or is focused around one of these areas, feel free to browse assays that are related to those research areas. Or if you have a gene of interest or a mutation of interest, you can feel free to search, use the search function. That's shown on the right. You can search by gene name, RSID, ClinVar nomenclature, or OMIM nomenclature, to name a few. And that can take you to your assay of interest even quicker. And lastly, how to go about, if the applications are in line with your research needs and we have the target of interest for you, how do you go about contacting us? So in the beginning of the presentation, we kind of called this out, but in the US, we sell direct through Canon BioMedical. So phone number, email address is shown here. So contact us at canon-biomedical.com or 1-844-CANON-BIO. For a phone number. And then if you're outside the US, we have a growing distributor network. So if you see your country here, the distributor is listed next to it and their website. If your country is not listed, we are continuing to grow our distributor network. So feel free to reach out to us at the contact us email address, and we'll be happy to get back in touch with you with kind of an update of new countries that we bring online. So I have spoken for way too long, and we wanted to open it up, make sure we had enough time for questions from the audience. So with that, let's open it up and see if there's any questions.
Thank you, Ryan, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many questions that we have time for. All right, let's get started. Our first question is, you showed Coriell controls being used in the copy number analysis. Can I use my own bank samples that have already been characterized as controlled instead?
That's a very good question. Absolutely. So we had, when we were doing the development of the assays, we wanted to make sure they worked with a third-party control. So if a lab didn't have bank samples away with characterized copy number changes, that they could, still bring in the assay and use some readily available Coriell controls. So that's why we had started there. But absolutely, if you have characterized samples tucked away or have access through a colleague of yours, feel free to use them as your controls for the copy number assay. They will work absolutely fine.
Great. And we have time for one more question, and this one feeds off of that. It says, do you offer this as a service?
We actually get this question a lot at trade shows and when we're out at customer sites. We don't offer this as a service. So the thought here was to put together a genotyping method that researchers who want to kind of bring this into their own lab or have it done more quickly than a send-out service, that they would have access to do so. So unfortunately, with the data that you saw, with the target that we're going after for the applications that we've touched on, we don't do that testing in-house, but we are happy to help bring your lab, bring it to where it needs and help out from a experimental setup perspective, and we can help out with the data analysis to bring these types of applications into your own lab.
Wonderful. I would like to once again thank Ryan for his presentation. I would also like to thank Canon BioMedical for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.